Welcome to Computational Biology. Today, we're diving into the fascinating world of functional genomics. If you have ever wondered how a single set of DNA instructions can create so many different types of cells in your body, you are in the right place. We will explore how scientists decode this dynamic biological story. Now that we know our topic, let us start with a simple analogy. Imagine you are given a massive box of Lego bricks and a printed list of every single piece. Knowing the exact list of pieces is like structural genomics. You know what materials you have, but that list does not tell you how to build a spaceship or a castle. Functional genomics is like watching the master builder actually use those pieces to construct something amazing in real time. It tells us when certain genes are turned on, where they are active, and how they work together. Instead of just reading static DNA letters, we are finally understanding the dynamic story they tell. Building on that intuitive idea, let us look at the formal definition. Functional genomics is a specialized field of molecular biology that uses vast amounts of data to describe gene and protein functions. Unlike traditional approaches that study one single gene at a time, functional genomics uses high-throughput techniques like RNA sequencing or microarrays to look at thousands of genes simultaneously. It encompasses subfields like transcriptomics, which studies all RNA transcripts, and proteomics, which studies all proteins. By analyzing this massive scale of data computationally, scientists can figure out the dynamic aspects of an organism, bridging the gap between the static DNA sequence and the complex behavior of a living cell. Understanding this dynamic behavior leads us to ask, why does functional genomics matter so much in the real world? If we only knew the DNA sequence of a human, we would not know why a liver cell looks and acts completely differently from a brain cell, even though they share the exact same DNA. Functional genomics allows us to see which specific genes are awake in the liver and which are awake in the brain. In medicine, this is revolutionary. It helps us compare a healthy cell to a cancer cell to see exactly which genes have malfunctioned. Without this field, we would be blind to the actual causes of many genetic diseases and unable to design targeted drugs. To see exactly which genes have malfunctioned, we need a way to measure their activity. When we measure how active a gene is, we look at its expression level, which is basically how much RNA it is producing. We calculate a metric called fold change to see the ratio of these expression levels. In this formula, FC stands for fold change. The variable ET represents the gene expression level in the treated or diseased sample. The variable EC represents the expression level in the normal, healthy baseline sample. If the treatment expression is higher than the control, the fold change is greater than 1, meaning the gene is upregulated. If it is lower, the fold change is a fraction between 0 and 1, meaning the gene is downregulated. Now that we have our formula, let us see this with a concrete example. Suppose we are studying a specific gene. Let us call it gene A, to see if it is linked to skin cancer. In a healthy skin cell, the expression level of gene A is measured at 400 units. This is our control baseline. In a cancerous skin cell, the expression level spikes to 3,200 units. This is our treatment condition. We plug these numbers into our formula, dividing 3,200 by 400. This gives us a fold change of 8. Physically, this means that gene A is producing 8 times as much RNA in the cancer cell compared to the healthy cell. While a fold change of 8 is clear, biological data can scale exponentially, which brings us to a better metric. We usually take the base 2 logarithm of the fold change ratio, known as the log 2 fold change, to make the data easier to analyze. In this formula, LFC stands for log 2 fold change. We apply the base 2 logarithm to the ratio we just calculated. We do this because it centers the data around 0, making it perfectly symmetrical. If a gene's expression doubles, the LFC is exactly +1. If the expression is cut in half, the LFC is exactly -1. If there is no change at all, the value is 0, telling us the gene is completely unaffected. Let us apply this new logarithmic formula to our previous cancer gene example. We already found that the raw fold change for gene A was 8. Now we need to convert this into a log 2 fold change to standardize it for our computational analysis. We take the base 2 logarithm of 8. Since 2 raised to the power of 3 equals 8, our log 2 fold change is exactly 3. This positive value mathematically confirms that the gene is significantly upregulated in the cancer cells. Biologically, this tells researchers that gene A might be driving the cancer growth, making it a prime potential target for new therapeutic drugs. We have seen how to measure a change in expression, but this leads us to an important question. Is that change actually meaningful? In functional genomics, just seeing a change in expression is not enough. We must prove it is statistically significant. Gene expression naturally varies, even among healthy cells. A small change might just be normal biological noise, while a large change could be a critical disease signal. To confidently say that a drug worked or that a gene is causing a disease, we need a mathematical way to measure how unusual our observed expression level is compared to the normal population average. To measure that statistical significance, we use a standard tool from biostatistics. This is the z-score formula, which is heavily used in functional genomics to identify outliers. Let us break down the symbols. Z represents the z-score, which is our measure of statistical distance. The variable x is the observed gene expression in our treated patient. The Greek letter mu represents the mean or average expression in the healthy population. Finally, the Greek letter sigma represents the standard deviation, which measures how much the expression naturally varies in healthy cells. By subtracting the mean from our observation and dividing by the standard deviation, we find exactly how many standard deviations away from normal our patient is. Now that we understand the z-score formula, let us test it with a clinical example. Imagine we look at the expression of gene B across a healthy population. The mean expression level is 50 units, with a standard deviation of 5 units. In a patient treated with a new drug, gene B's expression drops to 35 units. We substitute 35 for x, 50 for mu, and 5 for sigma. Subtracting 50 from 35 gives us -15. Dividing -15 by 5 gives a final z-score of -3. This means the gene's expression is 3 full standard deviations below normal. Since any z-score beyond -2 is highly significant, this proves the drug successfully suppressed gene B. Having calculated these metrics, let us visualize how scientists look at thousands of genes at once. What you are seeing here is a gene expression heat map, a very common diagram in computational biology. The columns represent different samples, such as a healthy patient on the left and a diseased patient on the right. The rows represent different genes, labeled gene 1, 2, and 3. The colors correspond to the log 2 fold change we calculated earlier. Bright red indicates a gene is highly upregulated in the diseased state, while bright green shows it is downregulated. Black means there is no significant change. This visual tool allows researchers to instantly spot patterns and identify which biological pathways are malfunctioning. We have covered a lot of ground today, so let us summarize the most important ideas. First, functional genomics takes us from merely knowing the static DNA sequence to understanding how that sequence operates dynamically in real time. Second, by measuring gene expression levels and calculating metrics like log2 fold change and z-scores, we can pinpoint exactly which genes are responsible for diseases. Finally, this powerful combination of high-throughput biology and computational mathematics forms the foundation of modern personalized medicine. You now understand how we do not just read the book of life, but actually comprehend its meaning and mechanics. Great job!